So in this talk, what I now want to do is tell you about a very specific project that I've been involved with really since the beginning. So this is a project that I co-founded with another scientist named Daniel Promislow and a veterinarian named Kate Creevy, with the goal of really trying to understand the biology of aging in companion dogs or pet dogs living with their owners. So all of the information I tell you today, the data I show you today as part of the dog aging project, these are not dogs in the laboratory, they're not kennel dogs, these are dogs living in the real world with their owners. And for me, this project, I think, really encapsulates a unique and powerful approach to one of the biggest challenges the field of geroscience or aging biology has been struggling with for certainly the last 10 years. And that challenge is we're really good at slowing aging and increasing lifespan in laboratory animals. If you saw my talk earlier, I showed you a bunch of data. We're really good at doing that in mice and in simpler organisms. But of course, laboratory animals, mice, flies, worms, don't live in the real world, right? And the human real world is very complicated. And so how do we start to test some of these principles, some of this knowledge, some of these interventions in a real world setting? And there are lots of approaches people are taking. Clinical trials are one. If you stop and think for a minute, let's just say you had a drug or a supplement that you believed slowed aging and could increase lifespan. And you had unlimited resources, so you could pay for the clinical trial. How long would it take you to do a clinical trial to show that you can increase lifespan in healthy people? Take a really long time, a couple of decades probably. So that's a hard thing to do in humans. There are people taking smart approaches. I told you about the work that Joan Manick did, looking at immune function, vaccine response. You can look at functional measures of aging, but you can't do a normative aging lifespan study in humans. It's not practical. So it occurred to me about 10 or 15 years ago that our pets are sort of a unique opportunity to both understand the biology of aging in the real world and potentially do something about it. So again, if you saw my earlier talk, I told you about my dog Dobby. One of the great things about studying aging in dogs is I get to show pictures of my dog, whenever I want to, which is a lot of fun. So you'll also notice, so I think dogs are a unique sort of bridge between studies of aging in laboratory animals and studies of aging in people. And you'll note I've got a double-headed arrow here, because to me, and I think to everybody on the Dog Aging Project team, one of the important things about this project is we're going to learn a lot about human aging, because dogs age very much like people do. But maybe more importantly, we're going to learn a lot about dog aging, and we're going to hopefully be able to do something about it and help our pets live longer, healthier lives. I've always been a dog person. I love my dogs. I love Dobby. And it is super important to me that we do everything we can to help give our companion animals the longest, healthiest lives that we possibly can. So that is, to me, more important than doing this in people. But we're going to learn a lot about human aging too. Okay, so there are really two goals to the Dog Aging Project. One goal is to understand the biology of aging, and the other is to do something about it. And I'm going to first talk about how we're approaching the first goal, which is through a large-scale longitudinal study of aging. I'll tell you more about that in a minute. And then at the end of my talk, I'll tell you about how we're trying to tackle the second goal. Before I dive into the meat of my talk, I do want to mention the Dog Aging Project team is enormous. So this has grown from, the three of us, Daniel, Katie, and myself, to a team of now more than 100 people at probably 20, 30 different institutions around the United States. It's a diverse, multidisciplinary set of scientists, veterinarians, clinicians, statisticians, all working together with this shared goal of understanding aging in dogs and doing something about it. This is a picture from our annual meeting in 2022. There I am with my Dog Aging Project t-shirt on. I should have worn it today. Let's see. Trying to find Daniel and Katie just to point them out to you, but I'm not seeing them right now. They're in the picture somewhere. There's Katie right there. So it's a fantastic team, and I just want to recognize the work that all of the team has done. Okay, so one of the things that people often ask is why dogs? What makes dogs such a great animal in which to study the biology of aging? And I think there are a bunch of reasons. As I've sort of already alluded to in my prior talk, dogs age biologically very much like people do, but they do it faster, right? About 7 to 10 times faster, it turns out. And again, I'm sure all the veterinarians in the audience or any of you who've ever had an old dog will recognize dogs get most of the same functional declines and diseases that people do as we age. It just happens faster in dogs. So biologically, even at the molecular level, it seems as though dogs are aging very much like people do. So we can learn a lot about the biology of aging in the real world because dogs share our environment. Dogs have this very unique genetic architecture. So again, everybody here I'm sure is familiar with the idea there are a couple hundred purebred breeds of dogs. So all the same species, right? But different breeds, kind of like inbred mouse strains. But then on top of that, we have this mixed breed population. And so that genetic architecture combined with the fact that dogs are very phenotypically variable. What I mean by that is if you look at almost any trait, and you compare dogs to humans, the variation in that trait is going to be as great or greater than it is in people. Just think about body size and the difference between a Chihuahua and a Great Dane. So the combination of that genetic architecture with phenotypic variability makes dogs very powerful for mapping genetic traits onto phenotypes. It's a sort of a very unique animal in that way. I already talked about the environment piece, or at least I alluded to it. This is super important. We cannot mimic this in the laboratory. In fact, we do everything we can to control the environment in the laboratory. But of course, people live in this super complicated environment that changes by the day, hour, minute. And for the most part, our pets share that environment. Diet is the one place where there are some differences. Outside of diet, dogs really do share the human environment to a very large degree. So we can actually understand the role of environmental variables in health span and longevity. And it turns out that at least for longevity, estimates are that about 70, maybe even 75% of human longevity is environmental. So 25% genetic, 75% environmental. It's probably about the same in dogs. People love dogs, and I think this project engages people in the scientific process in a way that few other projects can. I certainly have seen that with our participants. And again, as I've already alluded to, our goal is to improve the quality and quantity of life for the dogs and for the owners. I should have told my joke when it came at the environment. You probably won't laugh because of the translation anyways, but I'll tell the joke anyway. So to illustrate the environmental variability that dogs experience, very much like people. That's what these pictures are meant to show. Just like in humans, there are rich dogs. So I don't know if you can see that. That's the dog. That's the doghouse. There are economically disadvantaged dogs. There are dogs that practice a healthy lifestyle. I'll actually come back to exercise in a few minutes. And there are dogs that maybe practice a less healthy lifestyle. And I have to always make the disclaimer that I did not put the cigarette in the dog's mouth. This is not my picture. Dogs, of course, do not typically smoke, but they do breathe the same air that we breathe. They usually drink tap water, right? So in many ways, dogs are an environmental sentinel for some of the factors that may actually impact human health and health span and longevity. Okay. So this is the structure of the Dog Aging Project as it exists today. The largest part of the Dog Aging Project is what we call the Dog Aging Project PAC. To become a member of the PAC, dogs have to live in the United States. I'll come back to Brazil at the end. This is one of my frustrations. I wish the Dog Aging Project was an international project. I would like to see that happen. Hopefully Brazil can do something similar and maybe join forces with the Dog Aging Project at some point. Right now, dogs have to live in the United States, but if they live in the United States, any dog in the United States can participate in the Dog Aging Project if the owner is willing to complete what we call the Health and Life Experiences Survey, so HLES. This is an extensive survey instrument that captures as much information as we really reasonably can about the dog's environment. That includes other dogs in the house, diet, exercise, how much time they spend outside, inside, what sort of surface they sleep on, almost anything you can think about in the environment. Thank you. That might be relevant to health. We try to capture in this survey. It's pretty extensive. Takes people about 2 hours to complete all 10 modules. Once owners do that, they are part of the pack. The dog is part of the pack. We ask the owners to provide a veterinary electronic medical record that also will have health data. We ask the owners about the dog's health history, but we want to get the actual data from the veterinarians if we can. About half the owners who join the pack are able to do that. So as of today, I think we're up to about 53,000 dogs and owners participating in the Dog Aging Project. So it's a very large study of aging, okay? Annually, the owners whose dogs are in the pack are asked to update the survey instrument and the medical records. That's what makes this project a longitudinal study. If we only looked at one time point for each dog, it would be cross-sectional. But we're doing this annually so we can follow the same dogs over time. And that creates a very powerful opportunity to identify risk factors early that predict disease, health outcomes, longevity later. Okay, so that's the largest group. Owners who are able to provide qualifying electronic medical record become eligible for these other groups within the pack. These are what we call the sampled cohorts. There is a genetics cohort that we call Foundation. Now more than 10,000 dogs. Every dog in Foundation has its genome completely sequenced. And we return that data to the owners, but obviously we use that to try to make genetic predictions. 1,000 dogs become eligible for what we call the precision cohort. This is really, you can think of it like a systems biology cohort, where we're trying to get deep molecular phenotyping on these dogs. So annually, the owners whose dogs are in the precision cohort are sent a kit that they take to their veterinarian, and the veterinarian will collect blood, feces, and hair. And we do things like epigenetics. So again, for those of you who heard my talk earlier, we look at the epigenetic signatures in those dogs to try to understand, can we tease out signatures that are predictive for what's going to happen to that dog in the future. We also look at metabolome, microbiome, things like that, okay? That's the systems biology cohort. Every year, we get additional biological samples. We have what we call the centenarian cohort. Centenarian means living more than 100 years. These dogs obviously do not live more than 100 years, but the idea is that we want dogs that are very old, much older than we would predict based on their body size. Again, body size, it turns out, is the biggest predictor of lifespan in dogs. Big dogs age faster than small dogs. You've probably heard me say that earlier. I'll come back to that. So centenarian dogs are dogs that live much longer than they are expected to. The idea being maybe there's something unique about their environment or their genome that allows them to live exceptionally long time. And then the last group, which I'll come back to at the end, is triad. This is the clinical trial, test of rapamycin in aging dogs. And I'll come back and talk about that at the end. We'll end up having 580 dogs in that trial once it's fully subscribed. Okay, so again, 2 goals. I'm going to now take you deep, well, not too deep, shallowly into the longitudinal study of aging. So I mentioned more than 50,000 dogs in the longitudinal study of aging in the pack. When an owner completes HLES, joins the pack, they get this nice little certificate. This is Sage, beautiful dog, our 25,000th member of the pack. You can see here the distribution geographically in the United States. It more or less matches the population density within the United States. So we're not super overrepresented for urban versus suburban versus rural. And I've talked about some of the data types that we are collecting. Again, because we know where the dogs live, we do get air quality and water quality information that is tracked by the federal government, which is actually quite powerful. Okay, so I'm going to show you just a few of the things that have come out of the data so far, but I want to emphasize it's really tip of the iceberg time. So we really started collecting data at scale in late 2019. So it's only been a few years, and we've built the pack up to the size that it is now over that time frame. So we're really just starting to dive into the data. Already a bunch of very, very interesting observations, but there's a lot more beneath the surface, way more than our team can actually analyze. And you can, just to give you a feel, again, this is the size of HLES, up to 5,000 questions almost across 10 categories. I mentioned the electronic medical records. I didn't mention the cognitive assessment yet. I'll come back to this. In addition to the large HLES survey, every year we get cognitive assessments on every dog. And this is a clinically validated survey tool. Again, some of you who are veterinarians may know of this tool. It's called CCDR. It's used to diagnose dementia in dogs or canine cognitive dysfunction, but it's a survey-based tool that's been clinically validated as a tool to diagnose dementia. I'll come back to that. We get this cognitive assessment every year in the dogs that are in the pack. Okay, so because we know we'll never be able to do all of the cool science that can be done with this data set, we are an open science project. Every year, we deposit all of the data that's collected into the Terra platform at the Broad Institute. It's a cloud computing platform. Anybody in this room, well, I should be careful. Anybody in this room who's not doing this on behalf of a for-profit entity can get easy, simple, free access to this data. You just go to the website, you click on the button that says data access. There's a very simple data use agreement. So if anybody in this room is a epidemiologist, computational scientist, bioinformaticist, just loves dogs, you want to dive into the data, you can do that. If you're at a for-profit entity, you can also get access to the data. It's a little more cumbersome. We're still figuring out exactly what you're going to have to pay the Dog Aging Project. Dog Aging Project is a nonprofit academic project, but it has a lot of costs. And so we want to get something back from for-profit entities that are going to use the data for for-profit purposes. Okay, so the data is open. We also have a biobank at Cornell where we store those samples that I talked about that get collected every year. So academics can also get access to the biobank samples to do additional research. So it's an open science, community science project. Okay, so now I'm just going to show you a little bit of data. I'm not going to take you very deep, but I want to give you a feel for some of the types of data that are in the project and some sanity checks to make us confident that the data is high quality. So first of all, just looking at the age distribution, again, it's open to dogs of every age. We're a little overrepresented, and this is from the first 28,000. So this is not all 50,000 now that are in. We're a little overrepresented for, older dogs. We would have liked to get more puppies. We've got more puppies now. I think the name of the project, Dog Aging Project, confused some people and they didn't think puppies were eligible, but puppies are eligible. We love puppies. We love very senior dogs too. So we have a pretty good distribution though, and we have a pretty good weight distribution. The weight distribution more or less matches the weight distribution of companion dogs in the United States. We have a pretty good split of male and female dogs, but we are highly overrepresented for sterilized dogs. So in dogs, actually, you can think of them as having 4 different sexes, right? Intact males, intact females, sterilized males, sterilized females. And it turns out sterilization, not shockingly, has an impact on age-related health parameters in both dogs and people. Hormones are important, as it turns out. So about 90% of the dogs in the Dog Aging Project are sterilized. Again, that tracks pretty well with the population at large. The intact dogs, as you might guess, are overrepresented for purebred dogs because many of them are used by breeders of purebred dogs. And we have about 50-50 pure and mixed breed dogs. So again, pretty good genetic distribution. If we dive into, and of course we have all this data, if we dive into the purebred dogs, we have it's close to a couple hundred breeds represented, but the breeds are weighted by the popularity of the breed. Again, not surprising. A lot of golden retrievers, a lot of Labrador retrievers in the purebred population. And by the way, when we do genetic sequencing on the dogs, it turns out a not insignificant amount of the time, owners think their dogs are purebred. Turns out they're not 100% purebred. That actually happened with Dobby. He was like 6% other stuff. So close to purebred. Okay. So this is sort of interesting. So this is now all owner reported. That's another thing I should say. The survey instrument, it's kind of self-evident, but it's worth saying explicitly. This is all owner reported data. Okay. It turns out owners think their dogs are pretty healthy. So this is what the owners said when asked about the general health of their dog. Most owners think their dogs are in excellent health, and then it kind of goes down from there. But when you ask them about specific diseases that their dog has been diagnosed with, there's a lot of disease diagnoses in this same population of dogs. So perceived health of the dog and having been diagnosed with a disease don't necessarily match up. This is something you just have to keep in mind when you're thinking about owner-reported data versus veterinarian-reported data in the health records versus laboratory quantified data from a laboratory clinical test. Different levels of confidence. So this is just showing you the sort of highest level of disease diagnosis. You can see lots of skin disorders, lots of trauma, lots of dental oral disease. I actually think this is a low-hanging fruit point for dogs. Probably most veterinarians would agree with this. Oral health, I don't know about in Brazil. In the United States, most owners don't do much for the oral health of their dogs. And with aging, dogs have a lot of oral health problems that can drive disease in other parts of the body. So, I'm not going to show you the data, but within each of these categories, we get additional information. If an owner says, yes, my dog has cancer, we then ask, where was the cancer? So we have additional data for each of these categories, subcategories, where you can get finer and finer detail. Okay, sanity check. We know walking speed, activity should go down with age. When we look at owner-reported metrics of walking speed as a function of age, we get what we expect. So that's good. If it went up with age, I'd be worried about the quality of the owner-reported data. What about disease? So this is kind of interesting. This is cancer, all cancers here. We could do this for individual cancers. This is just all cancers lumped together. So this is a semi-log plot. So what you've got is a logarithmic scale on the y-axis and age on the x-axis. This is just showing you that risk of cancer, because it's linear, risk of cancer in dogs increases essentially logarithmically, exponentially with age. Same thing's true in people. Okay, what you probably can't appreciate from the way it's plotted here is there's a very strong body size effect. So I have said a couple of times, big dogs age faster than small dogs. That's true for most diseases, most functional declines, and it's true for cancer. So if we now look at all of this data plotted on the same plot, you can see the slope is the same by body size, but the risk is just always higher for any given chronological age in big dogs, consistent with the idea that their biological age is greater than their chronological age. Does that make sense? Okay, so these are the kinds of very, very simple analyses, first principle kind of stuff you can do with a data set like this. What are some more potentially interesting, sophisticated things we could do? So I'd been sort of studying things like caloric restriction, intermittent fasting. I talked about rapamycin, which in some ways is a chemical mimic of caloric restriction. And honestly, I had been very unimpressed with the literature in laboratory animals on intermittent fasting and time-restricted feeding, or what some people call time-restricted eating in humans. That's this idea of shrinking all your meals into an 8-hour window or a 12-hour window. In mice, that doesn't really do anything to lifespan unless you also calorically restrict. So I was pretty skeptical, but it occurred to me that our data set is an interesting opportunity in a population that sort of naturally varies in how often they eat. Because some owners feed their dogs once a day, some owners feed their dogs twice a day, most owners feed their dogs twice a day. Some owners feed their dogs 3 times a day. Believe it or not, some owners feed their dogs as much as the dog wants to eat. That's called ad libitum. There's clearly a self-selection there because some dogs, if you feed them ad libitum, are going to blow up into huge blimp dogs. But some dogs do get to eat whatever they want, as much as they want, and they don't become obese. Okay, so we have this natural opportunity here to just ask a very simple correlative question. And that is, if we look at a bunch of different age-related diseases, are there any differences between dogs fed once a day and dogs fed more than once a day? And again, I didn't think this was going to work. I thought we would not see anything significant. It turns out there's a very, very strong signal. We looked at 9 different age-related disease categories. We also looked at cognitive function. Cognitive function isn't shown here. In all of the cases, the dogs fed once a day had a lower risk of having been previously diagnosed with that disease. Okay, I'm emphasizing that because, again, you have to think about the way this data was collected. This is owner-reported prior health history. So what we're asking is, for a dog that's fed once a day versus dogs that are fed more than once a day, what is the risk of having previously been diagnosed with this disease or condition? In every case, it was lower in the dogs fed once a day. In 6 of the 9, it was statistically significant. That's the ones that are in red. And in some cases, it was a very large effect, like more than a 50% reduction in relative risk. Okay, so that's real. Like, I've seen a lot of biological phenomena where I'm like, maybe. This is real. But it's not necessarily causal. Okay, that you have to appreciate. Correlation is not causation. Unfortunately, the media sometimes has a hard time appreciating correlation is not causation. So, could it be the case that once a day feeding causes slower biological aging and less age-related disease? Yes, that is one hypothesis that can explain our data. Another hypothesis, which I actually think is probably more likely to be right, is that dogs fed once a day are less likely to be obese, and we know obesity increases risk for all of these conditions. So, could it be obesity that's driving this? Yes. Could it be something else about the dogs fed once a day? Yes. So, it's a hypothesis-generating observation that we now can go back and test with more detailed experiments, more detailed data collection tools. And based on this study, we have expanded the information that we get on diet quality and quantity for the dogs in the Dog Aging Project. Because we'd like to know, are the dogs being fed once a day eating less or eating a different kind of diet? Okay. So that's one example of what I thought was a pretty interesting analysis. Another thing we're very interested in is cognitive function and dementia. I guess I forgot to mention it, but in that pack diagram with the overlapping circles, we have a dedicated study of dementia that we call the Brain Health Study. Within that study, we're interested in understanding what are genetic and environmental factors that influence cognitive aging and dementia in dogs. As I mentioned, some dogs do develop dementia. It's called canine cognitive dysfunction. It's not the same as Alzheimer's disease exactly. It's probably, in people, we kind of break up the dementias into different disorders and diseases. I think in dogs, they kind of all get lumped together in the CCD phenotype, canine cognitive dysfunction. So the Brain Health Study is about understanding what causes dementia and to what extent in dogs that don't develop dementia do they show age-related cognitive declines like we see in people. Okay, so it's a dedicated study. We have owner-based games. We ask owners if at time of death they're willing to donate the dog's brain to the project. Those that do, we do molecular analyses on the brains of dogs with and without dementia. So it's a pretty extensive study. Great team. We've actually brought in some experts in Alzheimer's disease, in neuropathology of Alzheimer's disease in people to work with us on this project to understand how similar at the neuropathology level is canine cognitive dysfunction to Alzheimer's disease. So one of the questions we could ask, though, is with this survey instrument, do we see evidence for age-related cognitive decline? Okay, I'm gonna get in the weeds just a little bit with the survey tool so you understand how it's used. So this is the CSLB, Canine Social and Learned Behavior Survey, based closely off of the clinically validated CCDR survey. It's been validated as a binary tool. What I mean by that is if a score is above 50, that dog is diagnosed with dementia. If a score is below 50, it's diagnosed as not having dementia or dementia not detected. So it's kind of a yes-no tool. We were interested in understanding, would we see changes in the survey score in dogs that don't have dementia with age? And so what's shown here is the decline in cognitive function measured by this survey tool in non-demented dogs as a function of age. And what I think you can appreciate is there's this very steep decline in cognitive function with age in dogs that would not be diagnosed as having dementia. So we think this is pretty good indication that you can use this tool to detect age-related cognitive decline. Okay, and then you can do some cool stuff with that. One of the questions that I was interested in is what happens to cognitive function as a function of age based on body size. So I have said several times big dogs age faster than small dogs, right? It turns out that there's sort of weak evidence in both people and dogs that big dogs might actually be protected against dementia. But it's pretty weak, and it's hard to tease out because big dogs age faster and die sooner than small dogs do. So we could look across body sizes at this score, this cognitive decline score, and ask, are there differences by body size? And it turns out there aren't. So this is the one clear case, in my view, where aging of the brain, at least as measured by this survey tool, does not seem to be accelerated in large dogs. There's no obvious protection, but it doesn't seem to be accelerated. And I think this tells us something potentially important about aging of the brain relative to aging of the rest of the body. And it also, I think, has a biologically plausible mechanism. So we know that the biggest determinant of body size in dogs is signaling through a pathway called IGF-1, insulin-like growth factor 1. The IGF-1 receptor gene variation accounts for much of the body size difference that we see in dogs. We also know from studies in worms and fruit flies and mice that animals with lower IGF-1 signaling live longer and age more slowly, except in the brain. IGF-1 insulin signaling in the brain is probably, at least to some extent, neuroprotective. And so the biology here actually lines up really well with what we know about some of these factors that regulate body size and regulate aging. So it's pretty speculative, but there's a clear mechanistic hypothesis for why aging in the brain at a molecular level, at a signaling level, might be different than aging in other parts of the body. Okay. So other members of the team have also done some cool questions, asked some cool questions related to cognitive decline and dementia. Like, when we look across all the other things we're measuring, which factors are most associated with dementia in dogs? And it turns out age is number one. Good. I would be worried if age was not the greatest risk factor for dementia. Again, for those of you who saw my earlier talk, you'll understand why I would have been worried if that wasn't in our data set. In dogs, there's about a 60% increase in risk of developing dementia every year. But when you get past age, it's kind of interesting. Exercise is number 2. That fits really well, I think, with the human data. The idea that exercise is protective against the risk of developing dementia. I mentioned sterilization. So here, there seems to be a slight but significant risk associated with sterilization, with being intact. It turns out that sterilized dogs probably live a little bit longer when you normalize for body weight than intact dogs, but the cause of death seems to change pretty dramatically, and that's a different study that also has come out of the Dog Aging Project. And then last on the list is this prior history of neurological eye, ear disorders. Again, there's evidence in people, right, that sensory perception or maybe lack of sensory perception can accelerate cognitive decline. That may be what we're seeing here. That's speculation. And this was cool. The New York Times has written several articles about the Dog Aging Project, and they got interested in this study and wrote up a nice article about the study. Okay, so that's the longitudinal study. Now I'm just going to finish up by telling you about the clinical trial. So again, test of rapamycin in aging dog. The goal here is really to answer the question, can we take the gold standard pharmacological approach to slowing aging and increasing lifespan in the laboratory and demonstrate whether or not it slows aging, increases lifespan, increases health span in companion dogs? And the rationale, if you saw my talk earlier, you have seen a lot of the data. The rationale is rapamycin's the best tool we've got. Maybe with the exception of caloric restriction, at least from laboratory studies. But again, if you think about trying to do a clinical trial in dogs of caloric restriction as a longevity intervention, it's going to be really hard to get owner compliance with that study. And it probably would be really hard on the dogs. So we definitely felt that rapamycin was our best bet here. And given how fast dogs age, we can get the answer in a few years. And I'll show you the specific way that we're doing that in a minute. Okay, so I'm not going to tell you a lot about rapamycin. I did that in my last talk. Discovered on Easter Island, slows aging in every laboratory animal when it's been tested. A couple of things that are important for the design of our trial is, as I told you both before, if you were here, you can start the treatment in middle age and still get benefits in mice. That's what we're doing in dogs. Was that to tell me I have 10 minutes? Were you telling me I have 10 minutes? It says 17. I'll do my best. All right. You can start the treatment in middle age. That's what we're doing. And in at least some tissues and organs, you can see an improvement in function. I also showed you in my last talk that in mice, you can take middle-aged mice, so the mouse equivalent of a 60-year-old person or an 8-year-old middle to large-sized dog. You can treat them for a transient period, 3 months in the case of mice, and get big effects on lifespan. So shortish transient treatments in middle age in mice are enough to increase lifespan. The reason I'm telling you this again is that's the rationale for the way we're doing the clinical trial. Okay, so we've done 2 safety studies so far. It's worth stating explicitly, when you're doing a clinical trial in people's pet dogs, it's very much like doing a pediatric clinical trial. People think of their dogs very similarly to how people think of their children. Safety has to be the number one priority. We were fortunate with rapamycin that we had data from mice and data from dogs where we knew the dose range where we thought it would be in the range to potentially have positive benefits for longevity or health span, but where we knew the side effect profile would be minimal and the risk would be minimal. So we were able to start with those doses. Nonetheless, we needed to do short-term safety trials in dogs to convince ourselves that this could be done safely. So the first trial had 24 dogs. All of our trials are randomized, double-blind, placebo-controlled clinical trials done to the very highest clinical trial standards. We're not asking for FDA approval, but it's done to the standards you would do these trials to if you were going after FDA approval. So in the first trial, 24 dogs, 8 got placebo, 16 got rapamycin. It was a 10-week treatment period. The dogs had to be at least 7 years old and between 40 and 110 pounds. Middle-aged, larger dogs that are aging rapidly. They also had to be healthy for their age. This is not a disease study, it's a study of normal aging. Okay. In the interest of time, I'm not going to take you through the data. We saw improvements in left ventricular function that by echocardiogram that look similar to the data that I showed you in mice. We also saw no evidence for side effects. So every week, the owners were asked, did you see any of these side effects in your dog? The owners didn't know. So I said it was double blind. It's actually triple blind. The owners don't know, the vets don't know, presumably the dogs don't know either. So there's nothing that's statistically significant here, different between the rapamycin and the placebo. There were a couple of things that owners reported, so we asked them open-ended questions. So we asked specifically about these side effects. We also asked open-ended questions. Have you seen anything negative or anything positive in your dog in the last week, every time they completed the survey? Two things owners reported. We did not ask them about these things. They reported increased energy in the dogs getting rapamycin. That's interesting. And they reported their dogs were more affectionate. At least 25% of them did. I don't know what that means, but it could reflect changes in cognitive function. Very speculative, but these were given up by the owners without being prompted. So interesting. Okay, so we did a second safety trial, similar results, no side effects detected, no clinically relevant side effects detected. Triad has 580 dogs when it's fully subscribed. We're about halfway there right now. Half rapamycin, half placebo. This is the dose. I'm happy to talk more with anybody who's interested. That's the dose we're using, given once each week. It's a 1-year trial with 2-year follow-up. Lifespan is the primary endpoint. Given the cohort of dogs and this length of trial, 3 years total, we are statistically powered to detect a 9% change in lifespan. Okay, that's the primary endpoint. But of course, we're interested in health span, looking at multiple secondary and exploratory endpoints. And I told you about the 2 preliminary clinical trials. This is the design. The only thing I'm going to say is the dogs come into the clinic. This is all done at veterinary clinics that the owners have to come to. We have about 20 clinical sites now, every 6 months for the entire trial period. The medication is given by the owners to their dogs. Okay, so that's the clinical trial. It's ongoing. I can't tell you what the answer is because it's still blinded. I do want to just mention, I think the Dog Aging Project has had a large impact on the field, larger probably than just the data itself. I mentioned open access data repository. We've got almost 100,000 people signed up for our newsletter, 50,000 people engaged in geroscience research, a lot of positive media coverage, which often science doesn't get these days. And we're starting to see biotech companies take the template from the Dog Aging Project and use it to develop drugs on the for-profit side. So Trivium Vet is a company out of Ireland with a veterinary formulation of rapamycin. Loyal is a San Francisco-based company that is developing a different drug target for longevity in dogs. And Pet More Time is a new company here in Brazil that I am the chief science officer for. There's a booth out there. If you're interested, you can find out more. Our goal is to take the lessons that we have learned from the Dog Aging Project and apply some of those to improve quality and quantity of life for Brazilian dogs and hopefully Brazilian cats and other pets at some point as well. Okay, so I'm just going to finish up 2 more slides on opportunities in Brazil that we are working on now. So I mentioned Pet More Time. There's also PAMEC, which is a partnership with Pet More Time, nonprofit, inspired by the Dog Aging Project in collaboration with the military police from 11 states. We are working on the agreements and plan to launch this study. This will be a research study in the near future. We are, at this point, designing a clinical trial intending to have 4 years of monitoring, again, for that statistical power that I talked about earlier. And we hope to generate evidence to support the veterinary use of specific interventions to slow aging, increase lifespan, and improve health span in companion dogs. And this is being done in Brazil, which I'm super excited about and very grateful to my collaborators and partners at PAMEC and Pet More Time for including me in this project. Very exciting. Okay, this is Pet More Time. Again, booth is out there if you're interested in getting more information. Our goal here is to make resources, education available to veterinarians that they can pass on to the owners to give more personalized approaches to health optimization in pet dogs in Brazil. So thank you. Sorry if I went over a little bit. It still says I have 9 minutes and 48 seconds. So happy to take questions if there's time, but if not, I'm going to be around tomorrow at the Pet More Time booth in the morning. So if anybody wants to come by and talk, I'm happy to talk with you there.